Okay, thank you, and hello everyone. Thanks for tuning in, and thanks to Dominique and Eglantine for the invitation to talk to you this afternoon. And they asked me to talk about some work that we've done on potato before this project started, really, but they thought it might be relevant to some of the other crops we're working on, as well as potato that we could take forward in this project. So, I mean, texture is an important sensory component along with appearance, aroma and taste, but it's very important for mouthfeel, as well as for cooking time, which impacts on convenience to the consumer. And of course, if a food crop cooks quicker, especially when it contains some volatile nutrients, it may maintain its nutritional quality better. And for this work on potato, we made use of a type of potato in a subgroup of Solanum tuberosum called Solanum foreca. So this group has a number of interesting differences from Solanum tuberosum that we've used as a comparator to try and understand these traits. For example, it has very shallow dormancy. It often has yellow flesh due to its carotenoid content. It has different flavor, generally preferred, and different textural properties. So it has a soft, dry, floury texture. I think you could call this mealy, which is generally liked by consumers, and also has a high intensity of flavor. And interestingly, it cooks in about half the time of a tuberosum type potato. And any sort of quantitative descriptive analysis or consumer preference trial we've done over the years, the forecas always come out on top. So you can see these in orange here are all forecas compared with tuberosum. This is sort of typical results. So we're interested to know what's the difference with this type of potato. So some years ago now, probably about a decade ago, we thought we would try and understand why it cooked so much more quickly. And of course, we weren't experts in food technology, but we worked with a group in the University of Bath in England, Professor Julian Vincent, who recommended this wedge fracture test and helped us to develop this for potato. And here you can see two tuberosum types, and you can see the much quicker foreca types here, and how the outer cortical layers of cells start to slough off very quickly on cooking. And if we make cubes of these potatoes, we can see this is reflected in the peak force and work done for this blade to penetrate the cube of cooked potato. So these two are forecas, and it requires much less peak force or work for the blade to go through this material than these two tuberosum types. So we could get some sort of quantification of the differences in texture between these two types. And according to the old literature, a lot of these textural differences might be due to the starch content of the potato, the nature of the starch or the amount of starch. And so we took cubes in the texture analysis from different parts of the tuber. We labeled these 1 to 7 and carried out our texture measurement. And we had a mixture of tuberosum types and foreca types. And we didn't really see any significant differences between these cubes, including the ones from the pith, which has much lower dry matter content. The only difference really was this stem end cube 7 in these graphs, which tended to require more work for the blade to penetrate, i.e. they had a tougher, firmer texture. And we think this is because this is where the phloem strands all come into the tuber. It's got a high concentration of vascular tissue, which gives it a sort of fibrous toughness. And so, as I said, when we measure the dry matter in these cubes, despite there not being any difference in our textural measurement, we can see cube 4, the pith cube, has much lower dry matter. So we thought that the textural differences or differences in cooking time were due to something else. About this time, we were starting to do gene expression studies using one of the first versions of a microarray we had for potato. And of course, now we do RNA sequencing. But here we could look at simultaneous expression of about 10,000 genes in a potato. And these graphs just show the expression of 2 genes that we picked out from the data sets that are to do with cell wall biochemistry. So in green, we have this pectin methyl esterase, and these are 3 stages of Foreca tubers, going from early tubers, early developing tubers, to mature tubers in 2 Forecas compared with 2 tuberosum. And what we can see, just looking at the pectin methyl esterase, is that there's a much higher level of gene expression of this particular pectin methyl esterase in the tuberosum types and the Foreca types. This wasn't the case for all pectin methyl esterases. It's a gene family with quite a few different members. I think it's about 12 in potato. But this particular one seemed to stand out as being much higher in the tuberosum type and in the Foreca in the tuber. So PME, what it does, this enzyme, it catalyzes the demethylation of pectin molecules, which are a major component of the cell wall. And once it's demethylated, this is negatively charged now, and it can cross-react with calcium. And this forms this egg box structure, which gives a much firmer structure. So when you demethylate the pectin, you cook the potato, calcium's released from the cells, and it can form this firm structure. So if we get high PME activity, we demethylate the pectin and we get a firmer structure. And so we looked at the PME activity in our cubes. We went back to our cubes, made some more, and we then analyzed the pectin methyl esterase activity. And these are 3 Forecas where we see much lower PME activity than in the tuberosum type. So we thought this was consistent with the gene expression that we saw. And the cubes are all pretty similar in the PME activity. And in fact, if you look at the correlation between the PME activity and the work done in the texture analysis, all these are Foreca samples, which show quite a nice correlation, especially if you remove the troublesome cube 7 with the vascular tissue. So then we went on to see, can we see these differences in methylation in the cell walls that we might expect if there's differences in the pectin methyl esterase activity. So we made cell walls from these tubers and looked at them using FTIR. And the chemists tell us that this signal difference here is due to methylation of pectin. And in fact, if you look at the methanol content of cell walls, it's lower in the tuberosum types here than in the Foreca types, as you'd expect if the tuberosum types were all demethylated. So that all seemed to fit together. And so one of the things that we do to prove our hypothesis about gene function is to change the expression of the gene in a transgenic plant. So we downregulated the expression in these antisense lines or overexpressed it in these called sense lines. So these have more expression, higher level of expression and a higher level of activity of the PME, whereas the silence ones have reduced, although it's not complete, fairly small effect. But we can see this is reflected in the FTIR signals, as we'd expect in the transgenic lines, and the methanol content, particularly in the overexpressed lines, we see reduced methanol content, which is what we'd expect if we increase the PME activity. And again, more prevalent in the sense lines, we can see an effect in the textural properties in the tubers from these transgenic lines. It's not really significant in the silence lines, but there's a trend towards that. So that all seemed to make sense and fit, but from a breeding perspective, you really want to be able to use this to breed a potato where you might want to control cooking time. So can we find the gene variants that would be of benefit in this sort of work? And so we looked to see if there was a naturally occurring genetic basis for the difference between the Verecas and tuberosum. So for this, we made a diploid population of potatoes using a Vereca parent and a tuberosum parent. And basically, we ended up with a population of about 125 individuals that show quite a wide range of traits, shapes, and flesh colors, but also textures. And so we could do QTL mapping, quantitative trait locus mapping. So basically, if you measure the traits in all these 125 brothers and sisters, we have a dense genetic map. So we can see where the trait maps onto the chromosomes. And because we've got the genome sequence, we could see any candidate genes in this region. And so when we measured the texture QTL, we find it maps to exactly the same locus as the pectin methylesterase gene called Pest1. I think it's on chromosome 7. So there's allelic variance, there's variation in this gene that could be used to breed lines with different cooking properties. And this is what we're currently doing with collaborators by trying to integrate these lines into diploid potato. So that's really where we're up to this work. I was very interested to read the review from the RTB potato group. It's a very interesting read, and it did show in this paper that mealiness is an important trait for consumers in their studies from Uganda, and also other recent work agrees with this. So I'm just wondering if PME is involved in mealiness. It's kind of difficult to quantify that, but also be interested to know people's views on the importance of cooking time to consumers. And so I'll leave that question up there and just acknowledge colleagues who did this work with me in the James Hutton Institute, and also the work of the RTB Foods potato team. So thank you very much for your attention, and happy to answer any questions. Thank you.
Thank you very much, Mark. Thanks a lot for this description of the whole scientific approach to go from the understanding of biochemical determinants of texture up to the identification of QTLs or markers for texture. And it was a very clear presentation with very well chosen illustrations and meaningful charts. So thanks a lot. So feel free to ask questions to Mark. If you want, you can write them down in the chat box. Before Dominique's question, there is one from Emmanuel Alamu from IITA Nigeria. Emmanuel is asking, what was the sample size of each cube? So the number of data points for each cube, and how many tubers have you been using?
Yeah, that's a good question. All these details are in the papers that we published this work in, and to be honest, I can't remember. We did this work about a decade ago, but it's we use quite a lot of replication because there's quite a lot of variation in these traits. So I think we used about 6 or 8 replicates per data point, but this is all in the papers that I would have to look up. but I can share those with you if you would like.
And how many tubers to reach the conclusion?
Yeah, that's a good question. I think we probably needed about 3 or 4 tubers per data point.
Okay. Thanks, Mark. If you want to share the publication, you can share it in the chat box later or by email, and I will share together with the presentation.
Okay.
Dominic, you have a question?
Yes, thanks, Mark, to lead this discussion. For us, potato is a model plant. What about your recent work on sweet potato?
Sorry, Dominic.
For us, potato is a model plant because it's maybe the roots that we have more information at the moment. But what about your recent study on sweet potato?
Yeah, I don't think it's quite the same in sweet potato. So, but I think some of the other, I think starch might be more important in sweet potato than it is in potato. Some of the data that we've seen from Christian indicated that, and also the breakdown of starch. I think some of the work is indicating that beta amylase might be important. So I think it might be quite different, but we've now prepared cell walls from different sweet potatoes with different cooking properties and hope to have those analyzed fairly soon. So we may be able to answer that a bit more clearly in the coming weeks.
Good news. Thank you.
Another question for Mark. There are people working on sweet potato and potato attending the call from SIP Uganda, SIP Kenya. Is there somebody who wants to add something to Mark or make a comment on his presentation?
Okani, Mariam.
No questions, no remark?
Okay, this is Xiaofei from Seattle, the Cassava breeder. Can you hear me?
Yes, we can.
Yeah, I have a question about, so this is potato, but so we can see dramatic difference between the two types of potato. So in your QTL mapping study, so that QTL is the only major QTL you find, or there are still some other QTLs? And another thing is how much percentage of variation this QTL can explain, and do we have marker available from this QTL used for breeding?
Yeah, we're sequencing the alleles of the variant, so we have the actual alleles, not just the markers. It was the major QTL for cooking time that we detected. I think it explained 15 to 20% of the variation in that region. There were a couple of other minor QTLs, but of course, you know, the trait is quite difficult to measure. So
yeah.
you don't get, you know, there's quite a lot of noise on the analysis.
I agree. Yeah. Okay, thank you.
Thank you for the question.
We have a question from Aliénor from INRAE, Avignon. I will read it. It's in the chat box, by the way, Mark.
Yeah.
The PME activity can only be assessed on fresh vegetables or cold powders. Because of the epidemic, we received fried samples for the moment. Can we use the methylation degree as our only indicator?
I don't, I'm not really sure if that's been tested. I wouldn't have a great deal of confidence until there've been some validation. Sorry, not very helpful. But there may be there may be people who would know the answer to that that question better. But yeah, I'm not sure. I'm sorry.
Okay, thanks. Thanks for trying. There is another question from Wasiu from Nigeria. What could be responsible for the changes in texture of different roots and tubers with reference to season of the year?
Oh, yeah, there's always the G/E effect, but I mean, I think the PME effect is fairly heritable, so it is a strong genetic effect. Yeah, I'm sure there'll be environmental effects of on these cooking times that will moderate any measurements we make. I mean, that's what you find with virtually every trait we measure in potato.
Thank you. There's a question from Suzanne. What are your thoughts on mapping sensory texture traits to QTLs rather than instrumental measures of texture? So mapping sensory.
That would be very interesting. So if you use a QDA approach to measure mealiness, for example, that would be very interesting to know where that trait appears. But it's I think it's quite difficult to map QDA data. I know we tried to do this quite a lot, and we had a big attempt quite a few years ago to do this, and it's quite ambitious to map QDA data. I think the instrumental data is probably more robust, but if you've got a strong effect, it might be possible to map it, and that would be a very strong indication. So, for example, mealiness in potato would be fantastic to map, and it'd be even better if it mapped where the PME is located.
Another question to Wasiu to complement the first one. Is there any effect of G/E on the pectin content of potato as it affects the texture?
We don't have that depth of data, so it'd be interesting to look at that perhaps. But you know, these are not high throughput measurements, so you don't undertake these experiments, you know, without some quite a lot of resource to help. So it would be interesting. I'm sure there's temperature effects, for example. Different environments would affect PME activity. I'm sure, and expression.
Another question. I don't see any more questions in the chat. Dominique?
There is one from Suzanne. I don't know, you don't see that. What are your thoughts about mapping sensory texture traits to QTL?
Yeah, I answered that. It was about, you know, can you map mealiness onto, it would be very interesting, but difficult. But I think that's a good question and might be worth trying.
Do you know some work on that?
I don't know. It's something we tried, but we never really got good enough data to publish. We tried that in potato.
Okay.
I don't know if anyone else has tried it in other crops.
Some more comments? Remarks?
What about the microscopy? Can we see something with microscopy of the cell walls? Are there any elements that we can have on the microscopy?
Yeah, so I mean, there's quite a lot of well-characterized antibodies that pick up different pectic epitopes. So you can use those to look at the cell wall structure, and you know, can be quite instructive. So there's certain antibodies that will recognize methylated pectin, for example, or different pectic epitopes. They can be useful. It's something we tried to some extent, and we did. Yeah, I think that could be interesting, worth trying in other crops apart from potato. Yeah, for sure. Cassava, sweet potato. I'm unaware of any work in that area, but that could be an interesting approach. If you had, if you had the technology to do the immunogold labeling.
One last question, maybe from the audience before we close the call. I saw a comment from Yusuf, but I could not get the question, or whether it was a question or just a comment. So if Yusuf wants to.
Please.
Pathway analysis.
Yusuf, are you with us? Yusuf Mukasa? Yes. Can you open your mic and ask your question or make your comment directly to Mark?
You are mute.
Apparently, Yusuf has troubles to unmute
Hello?
himself. Yes.
Yeah.
Hello?
Hi, Yusuf.
Good afternoon, bonjour. My question is, have you tried to do some PME mapping, enzyme mapping, to try to understand exactly how PME activity affects the texture traits in potatoes? Then I see you have used 2 varieties. Can you also try out with other varieties from other regions? because enzyme activity may also differ with location, geographical sites. So can you also try to do it with other geolocation, maybe Latin America, Africa, and Europe?
Well, I mean, we looked at the PME activity. I mean, basically, a lot of our work, has to be said, is based on this difference between the Foreca and tuberosum, which is quite large. You know, they're very diverse, because that makes it a lot easier to understand the differences. So differences within tuberosum may be more difficult to actually pick out. And yeah, that's a good question. We don't know if the differences between tuberosum varieties would be solely due to PME, or whether it's, you know, just the difference between the Foreca and the tuberosum. So, and we haven't really explored that differences within tuberosum, but virtually every tuberosum we look at has much higher PME activity than the Foreca, and we can breed the Foreca alleles into tuberosum. So, you know, it's not really something that we're too concerned about, the differences within tuberosum. But I think that's, if that's the point you're trying to address, I think that's a good point. I don't know if I've answered your question correctly. Is that what you were trying to say, Yusuf?
Yes, you have answered it well. Thank you very much.
I see a question from Emmanuel.
Yes, thank you very much, Mark. I just look at the cooking of the potatoes, the biochemistry, what happened with the PME when it is demethylated, and then the calcium play a role to make the hardening. So in essence, the calcium content of the potato play a good role in the cooking time. So I just want you to comment on that.
That's yeah. I think. Sorry, you know the calcium does. I think calcium does influence cooking time in potato. Is that something that people have tested? I would predict that it would. If you have
people have tested in pasting property. You know, adding different salts like calcium chloride, and it has effect on the viscosity, especially initial and the final viscosity. So I'm just looking at the pathway that you have given. That means it's a thing for us now to look at when you are studying the cooking time, that the calcium play a major role in forming the hardening of when the PME is demethylated.
Yeah, and I think if you collated the calcium with EGTA, you would increase the cooking. You'd make the potatoes cook quicker, perhaps.
Yeah, I just bring it out for us. You know, we are looking at the cooking time, really, and then I still believe that we still need to look at the calcium role in forming the hardening. So how quickly it is. So that's why I just mentioned it, that this is just part of the factors that we need to look at, as well as not only the PME and then the PME activity. You look at the role the calcium salt play in making the hardening when you are applying the temperature you are cooking it. Thank you.
Yeah, that's a good point. Yeah.
Thank you, Emmanuel. Other question? Comments, remark, suggestion, whatever, before we close? Mark, do you have a final word?
Yeah, well, I think we've still got a lot of work to do to understand these traits, and yeah, it's not easy trait to handle, but I think we have made some progress, and hopefully we'll understand this trait a bit better in all the crops before the end of the project. So yeah, I look forward to seeing what results we get when we do all these analyses that have started.
Thank you, Mark. We are also looking forward to see preliminary results applied on other crops. And thanks a lot for this presentation. And Dominique and I would like to thank you very much for agreeing to present these results at the very last minutes, because we were struggling in finding a presenter for this webinar. So thanks a lot. And we will meet in 2 weeks. And in 2 weeks' time, we should have another presentation on the role of parietal compounds on processing ability of yam tubers, if I'm not mistaken. Aliénor and Agnès Roland-Sabaté from INRAE Avignon will make this presentation on the role of pectins and so on, applied to yam tuber, if I'm not mistaken. But we will provide you more details as soon as we have a title for this presentation. So in 2 weeks on yam, pectins on yam, and in a month's time, it will be on plantain preferences and quality traits in Nigeria. And it will be a presentation made by Delphine Ama, which is the product champion for fried plantain in RTB Foods project. So thanks everybody for joining today, and we will share the presentation, the recording and the DOI to access the papers mentioned by Mark on our Slack platform. And I have shared the link with you, the invitation link to join our Slack platform in the chat box. For now, the material, the presentation haven't been uploaded for sure. I will in a few minutes. And so you will be in capacity to see, to access the DOI.